This is episode 90 of the Immunology Podcast, explaining immunology with doctors JoAnne Flynn and Stephen Hedrick. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Doctors JoAnne Flynn and Stephen Hedrick on the podcast to talk about their research and their work with the American Association of Immunologists and the AAI's Immunology Explained initiative. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Immunology 2025 is the world-leading annual all-immunology meeting, and this year it's in Honolulu, Hawaii. Join us from May 3 to 7, 2025 to connect, collaborate, advance your career, and access cutting-edge immunology research. Abstract submission is open through November 19. Learn more at immunology2025.aai.org. Hey, Jason.
Hey.
What up?
Have you been following the Nobels and something near to our heart for some recent coverage we've done?
I have to admit, most of what I heard is like the... AI, like machine learning models, and I forgot what it was. I was looking at the list again.
It was for proteins folding.
Of course. Yeah, well, isn't the authors of the Google platform for protein? I forgot the name.
DeepMind is IBM, I think.
Oh, yeah, Google DeepMind. Yeah, I was right.
No, it is Google.
Yes. So AlphaFold, basically.
AlphaFold, yeah.
Okay. Yeah, that seems very, I mean, I guess it deserves a Nobel Prize. It's been quite a revolution. But it's interesting because everybody here works at Google, that right? Or worked or did this work at Google. So now companies are getting Nobel Prizes, I guess.
Yeah, it is. It's interesting. I mean, they were an academic originally, but...
Yeah. And then what else? Also, physics also got some, also in AI.
It's the year of the robots.
They're taking over. Is this the Nobel, Council getting, in the good side of the robot revolution? Is that what they're doing?
Probably.
Is that what they're doing? But well, of course, one of the comments on the socials was that, again, for another year, every single winner seems to be a man. So yay for like the 4% of women laureates. We still have some way to go, I guess.
Yeah, I mean, it takes time to be, if there's been a barrier, it takes time for, even if the barrier is removed, to propagate to that stage in the career where you get the Nobel, right?
Yeah, I guess. Still.
I mean, it's like, 60% of college attendees and graduate are female now, but you won't see that downstream through PhDs and to Nobel Prizes for like 40 years.
Yeah. Well, I mean, these people were not too old, but in any case, what I do think it's in biology or medicine, physiology or medicine, that was for was it microRNAs or
microRNAs?
I would have thought that would have already gotten a Nobel Prize by now. So I guess that one I think was overdue. No?
Yeah.
We've known about these guys for a while, about microRNAs.
Yep. No, it's good times. All right. Well, we got to get on to science land on our end here with other things. So this is in developmental cell. It is sex-biased human thymic architecture guides T-cell development through spatially defined niches. First author is Laura N. Stankiewicz. Last corresponding authors are Peter W. Zanstra and Fabio M.V. Rossi. Coming out October 8th. All right, so this is one of those papers that is somewhat hard to describe because there's a lot of pictures and, this is an audio presentation. So what they did is they took postnatal thymus from babies who had to undergo cardiac surgery and took the thymus as a clinical trial, as was being involuted from volunteers, and then did multidimensional sequencing and multidimensional imaging and multi-omic sequencing. And I can describe it a little bit. And then looked from the sequencing at receptor ligand interactions. And then from the imaging, they had niche compositions and also could see some of the receptors that were touching each other, right? Because you're going to have some fluorescence imaging of major receptors next to each other, and use that to establish niches and understand what was going on, both from a niche perspective and a sex perspective for sex differences. So they use something called codex imaging, which is DNA barcoded antibodies to then have multiple colors. And so they have that. And on top of that, they then have site-seq and attack-seq going on to get you all of your data. And then they describe it all for a very long period of time in all of the detail. I think in the end, you can imagine this paper has some pretty cool pictures, a lot of omic analyses. I think I want to distill it down into key points. And one of these is a little bit of a ding on the presentation here, because I was going into this thinking, there'd be some really cool sex differences that I was going to talk a lot about, and this is really going to be about sex, but I really just baited you when I said the name of the title, Brenda, because there's some sex differences, but it didn't stand out to me. What they do is they're able to find a lot of different, these different niches in the thymus that they describe in pretty good detail that combines with what we know, but they're able to track the selection of immature activated T cells that are, CD4 and CD8 positive, and track that through. And also they're seeing different Treg populations they didn't appreciate before in the selection of both of these. So that's one cool theory, in different centers within the thymus, close, near the corticomedullary junction and the cortex, back in the medulla. So in the different spots, they're able to see this and see the signaling there that aligns with what we've understood today, but in a much more high resolution. And then understanding cells, and then subtypes of cells and the progression that they kind of leave as unknown, and they're not really don't really know the trajectory or some other stuff. So there's a little bit of work there. So there's that. The second little bit of this is then they say sex differences. So in increase in females, they see more HLA2 class 2 self-selection. So that's CD4, right? And more JAG interactions. And in men, they see more of everything else. IL-1, receptor 2, CD40, TNF, VEGF-A, FGF expression, proliferation, prostaglandin signaling, more inflammation stuff. What this means, they don't describe. How profound the effects are, unclear. Meaning of the effects, So I was really kind of going in, maybe this would explain some of the downstream differences in sex-based immunology. But obviously it's hard to deconvolute that from all the, differences in sex hormones we have flowing through our bodies the rest of our lives. Maybe there's something there with the HLA class 2 selection. Maybe not. I don't know. The authors don't know. So this paper is really cool if you care about the thymus. I think the technology here, which is cited from other papers as well, that they do is really neat for like exploring a tissue and understanding regional and sequence, and then what the cells are doing at each point in space. But also kind of wasn't what I was hoping for when reading it, when I'm like, there's going to be some really cool differences I'm going to find and some profound new thing in the niche. They're like, we have the niche. We see some cell types that we didn't know about before, or that seem to be doing some things, but we don't know what that means. Thank you. So there was no next level, like, because of all this cool omics imaging, I found this new thing out, right? They did all this omics and got a much deeper read on something than we've had before. So developmental cell, right? But it didn't get that next step, which is why I think it didn't quite hit cell, right, itself. It didn't have that next in-depth, like big aha light bulb moment from what they described that I was hoping to see. But still cool work.
All right. Very nice. Yeah, because I guess one of the main, always the question, we discussed it before in the podcast, right? That the higher incidence of autoimmunity in females, I think, is a very established phenomena. And I think we don't completely understand where that difference is coming. So we've touched upon this subject before. There's this certain immune complexes that are present in females. We have differences in the hormones, especially in adult females, and we know that here they're looking at more like more behind, like more basic thymus development differences that might also explain a immune system that is more primed for the immunity of some sort,
Right.
right?
So I was hoping
Yeah.
they'd be like, oh, see, here it is. And they didn't.
Yeah. Well, I guess we'll never know what a mystery it is to be a woman. All right. On that note, let's continue with our next big story of today. I want to start talking about tissue residency, and I'm gonna go. I hope I don't get anything wrong of this paper because it's quite a complex paper. So I was like at the last minute, I was looking at it and thinking, do I understand this correctly? So let's hope. If not, the authors, please do contact me or shout at me on Twitter, and I'll make sure to fix the record. But so the paper I want to talk about first is called Retinoic Acid and TGF Beta Orchestrate Organ Specific Programs of Tissue Residency. And this is a paper in Immunity. First authors Andreas Obers, and the last authors are Colby Zapf, Maximilian Efrard, and Laura McKay, which was a guest in your podcast some time ago. Um, and particularly, I'm familiar with Laura McKay's work to some extent. She has been working on tissue resident memory cells for a while. She has published one of the a few of the seminal papers in the area. So, coming from her lab, this is very interesting. Um, and so in this paper, so the question always is you have, particularly, I think most of the studies are done on CD8 cells. So this tissue resident phenotype, what does it mean? You have cells that after a immune infection, especially, most of these models are using like viral infections, herpes simplex viruses, LCMV, things like that. They tend to leave behind a population of cells that are permanently kind of stuck in the tissue. And most of the work has been done around cells in the epidermis or in the dermis. But we also, of course, know there are other areas that are very much studied for tissue residency. Besides the skin, we have also the liver. There's a lot of cells that are, in the parenchyma of the liver and are there. And also in the small intestines, there's also, they live within the tissue of the small intestine. And the idea of the tissue residency are cells that have, very more or less defined set of markers and transcription factors that characterize these cells. And these cells are very, the idea is that once they get in the tissue, they stay there and they get to survive in the tissue with the signals that are present in that environment. So they don't go back to the lymphoid tissue, they stay. And that's what makes them tissue resident. There are certain markers that are used to kind of identify tissue residency. When it comes to like surface markers, you have CD69, which is it's a surface molecule that inhibits the expression of SPR1 ligand, and that allows the exit of cells from the lymphoid tissues to the tissue, to the non-lymphoid tissues. And you have CD103, which is also a integrin that binds to tissue, that allows adhesion on the tissue, and that also really gives cements the location of a cell with outside of the lymphoid tissues. So one of the things, and just I think I need to give a little bit more background to understand why the models are using. One of the things that actually was one of the papers from Laura McKay, I think almost 10 years ago, they show that TGF beta signaling is important for the establishment of tissue residency. And one of the things that she showed is that there's a couple of transcription factors, TBET and EOMs, that need to be, so they're present in activated CD8 cells, and they're part of kind of the effector CD8 program. But in order for a cell to acquire tissue residency, they need this transcription factors need to be downregulated. And in the skin, and so they show in models of skin that this downregulation is achieved through TGF beta signaling. But, and this is important, they cannot be completely eliminated. So if you eliminate particularly Tbet, you have the opposite effect. You have, in the case of this is on skin, you have a loss of tissue resident cells in skin throughout time. So you have an initial seeding, but then the cells don't survive or they don't persist in the skin. And this is because you also need Tbet in order to uh, make cells responsive to the IL-15, which is the cytokine that have been shown to maintain the tissue resident populations in skin. And this IL-15 is made by tissue resident dendritic cells, and that's how this kind of niche is formed for these cells to reside. So this is why, but we know there's other mechanisms, because in the case of small intestine, they don't require IL-15 to survive. So basically, that would mean that they wouldn't require this residual Tbet expression to respond to IL-15, because they don't respond to IL-15 at all. So if you don't have IL-15, they are still there. And I think this is what kind of the idea that they were thinking in Lara McKay's lab. They were thinking, well, what are these other signals that are guiding the kind of the persistence of tissue resident cells in other places, such as the small intestine? And they also think of the liver, which is also independent of IL-15. Or, yeah. So, no, independent of TGF-beta. So basically, their first figure is you have Tbet deficient OT1 cells. They have a mouse model in which they infect with, I think it's LCMV, or HSV ova or LCMV ova. So, the ova is the target antigen of OT1 cells. And if you have a deficiency of Tbet, you end up in the end with, as expected, in the skin, there's less. There are less tissue resident cells in the end if you have complete absence of Tbet. This is what I think there's nothing very new about this. But in the small intestine, you actually have more. And so the question is, if not Tbet, what else is guiding this? And why do we have more? We don't have the same. We even have more than with Tbet. So basically, they do a lot of analysis. I don't want to go too much into the details, because I think let's just go on a little bit to the punchline. What they show is that this other signal that is guiding the increase in numbers is retinoic acid. So the main kind of connection here is that when they're looking at, they do ATAC-seq, or it's just like regular chromatin accessibility. I don't know. I think it's just regular chromatin accessibility assays. No, ATAC-seq. Yes, ATAC-seq. That and they show that basically there's what some of the access the sites that they see that are accessible, that are different in Tbet deficient OT1 cells, are those that are associated with retinoic acid signaling. So you have these RAR, RXR transcription factors that are responsive to retinoic acid. And retinoic acid, of course, is this metabolite that is we don't make ourselves. We get it from vitamin A, and then we process that into retinoic acid. But it has already been shown to modulate lymphocytes in the gut, particularly induces Tregs, modulates the conventional cells. So it's kind of anti-inflammatory, immunomodulatory. I think we already kind of knew that, and now we're seeing that it has an effect on tissue residency. So they do a lot of analysis. They have, this. They do. They trying to understand how is it competing with Tbet? Once Tbet is gone, why do we see this increase? And basically, they show that Tbet expression is controlling the retinoic acid, the effects of retinoic acid signaling through one of these transcription factors, RAR alpha. And basically, when you don't have Tbet, then you have this communication line is open for retinoic acid to drive these persistent. Basically, what they do in the case of small intestine, they prevent the exit or the egress of resident memory cells from the small intestine, and the cells, they stay longer, basically. And then you have this accumulation that you see in the end in the numbers. They also show that retinoic acid is important for the establishment of liver resident cells. So that's also very interesting. And basically, they want to show that you have, these different signals that are, they also interact with TGF beta, but there's this really different programs that either TGF beta or retinoic acid, they are both capable of imparting certain types of tissue residency programs. They do some in vitro studies in which, they activate cells in the presence of agonists or retinoic acid, signaling agonists or TGF beta. And they kind of show that there is this, that whereas skin resident memories seems to be mostly correlated to TGF beta signaling, the ones in the small intestine is kind of a combination of both. And then, the programs in the liver are mostly kind of retinoic acid derived. And this is interesting because when you think about the availability of either TGF beta or retinoic acid in different tissues, it would make sense because retinoic acid is, produced in the gut by microbiome, and then we also make it ourselves I think it's in the liver that we make it. We definitely store it in the liver. So that's why it's going to be retinoic acid in the liver. So it makes sense that, the signals are affecting tissue resident memory cells in the liver. Yeah, and then they have also these also interesting, kind of one of the last experiments. They have this dirty mouse, quote unquote. They are this mouse that have been co-housed with I think mice from like a pet shop or like wild mice with a wild microbiome, and which includes more bacteria that can make retinoic acid, can metabolize retinoic acid, and they show that they have an increase in the signal in this tissue resident generation in the small intestine that is kind of seems to agree with the idea that retinoic acid can drive this in the gut as well. So I think that's also very interesting. Whereas you would think that retinoic acid doesn't really get much into the skin, because it has to be transported by like dendritic cells or something. So that's a little bit less. The influence in the skin is less, and I think that kind of makes sense. So yeah, just basically you have this new ways of kind of new signaling pathways that are driving the generation of these tissue resident memories. It's not all TGF beta and TBET play. There's other components, and then retinoic acid, comes here as a new player in the field. So very interesting, very, very complex. I mean, I skipped through a lot of things here, but I think that's the gist of the publication.
Interesting. So retinoic acid inhibitors for like Accutane for acne are known to be exacerbate inflammatory bowel disease or cause it.
Yeah. Yeah. So I think that's also has been established that Tregs usually also can be differentiated with retinoic acid, and it definitely has immunomodulatory properties, in particular in the gut. So that's why you also want, those bacteria that are making retinoic acid. It's usually good to prevent inflammation in the gut.
Yeah, no, it's really interesting.
Yeah.
All right. So I guess I have to talk about COVID now.
Yeah. Well, if you must, go for it.
All right. So this is about a new super antibody that they're advertising for. It is a neutralizing antibody. So it's in Cell, a potent pan-SARS-CoV virus neutralizing antibody resilient to epitope diversification. First authors Lauren E. Rosen, M. Alejandra Tortoresi, and Anna DeMarco. Last authors are David Corti and David Vessler. So the way this group worked is they took people who were immunized against COVID 3 times and also had Omicron infection in 2022, and then took blood cells, got antibody sequences, they got antibodies, did cross-reactivity against receptor binding domains for SARS-CoV-1 across, and then did affinity maturation as well, and then used some machine learning guiding things to make it even better, and then tested it against vesicular stromatvirus, VSV-based pseudotype viruses to start with. And then they do a bunch of properties and measure its ability to neutralize and do, so they do mouse models or hamster models of infection, show that they work really well. They do some mutational analysis, so they'll mutate either the receptor and with known mutation patterns that have come out in the wild, so they're not making new mutations, just newer mutations since Omicron that have shown up that are resistant, and compare that and with with antibody docking models to the structure of the antibody that they got from cryo-EM, and try to understand what's going on. So on the one hand, it has a very wide activating antibody. It works against animal sarbecosis viruses, SARS-CoV-1, SARS-CoV-2. It has a high barrier, as they describe it, to viral escape, so less mutations work on it, which is true, and I'll get to that in a second. But they go through a list of known mutations and Omicron variants and other things and say, hey, look, this works against all of them. They look at fitness things. So, hey, mutation, I'll get to that. So the fitness thing is where I think it falls down. So I'm jumping ahead. But they basically show, hey, this has a pretty good binding region that binds to that receptor binding domain for entry on angiotensin and other parts of the virus too. So, right, it's not the angiotensin receptor binding part. It's the receptor binding domain of the protein that the spike protein is going after. But it's going after not just the ACE, because only COVID goes through ACE, but goes through other things. And so it goes to a different region and it binds pretty tightly there. And it does it in such a way that mutations are harder. And they show this structurally. And that's really cool. However, with mutational analysis and escape analysis, they find that, some of the mutations that allow escape have less fitness, which is good, right? That means the virus isn't going to live. And so it won't work. But they also said, hey, some of these newest mutations since the time that we first submitted to resubmission, they're already escaping this somewhat. They're having an effect on it. And so they talk about that's great. But then they then end up at the point where even the time between submission and resubmission, which was, received February 28th, revised July 1st, accepted September 16th, all of the same year. So not that far, right? 6 months, that even in a 6-month time period, some of the newer mutations are starting to escape it. Now, it still works pretty well in these, but it's already having less efficacy. It binds super tight, great, but binding less tight than it did before. And so I have mixed thoughts here. One, it does show a really good platform to finding good antibodies. I like this. It has a pretty quick cycle time, but it probably took them a year or two with all of this. And it does work really well. It's a pretty dang good antibody, better than what we have now. But it's already showing escape. And this is where I get to a question here. People get infected with these viruses over and over again because circulating antibodies at low levels are not sufficient to neutralize it and sterilize the infection. And this one shows it's relatively sterilizing early on as well. It neutralizes its ability to infect. And this is true because the viruses mutate in such a way frequently enough that our antibodies do a great job preventing us from dying because there's always some cross-reactivity, but you still get a cold all the time, right? Because antibodies aren't the key to fighting these off, at least IgG antibodies. I know we've talked about IgA nasal intranasal vaccination. I think that's a really interesting concept that I don't want to apply my statements here too. But IgG antibodies are not the mechanism of choice for our bodies to fight off these viruses. And I think we're tilting at a windmill here to try to get it to work as a therapeutic.
Yeah, Team IgA as well.
Yeah, so that's where I'm at.
Well, but I guess in this case, does it help with severe disease?
They don't do that. It just prevents the hamsters from dying. They don't look at the downstream mutations and they just say, hey, it's not working as well now. But we don't need antibodies to prevent severe disease, generally speaking.
Yeah. I guess you're right.
These neutralizing antibodies aren't for that.
No, they're not. What are they for then? Who would get neutralizing antibodies?
I'm old and I'm sick, so I'm going to take it immediately, not when
Oh.
And I'm old and I get COVID, I'm going to take this. But they don't really speak to that here, so I don't want to jump.
But would you take that? I mean, who would you have to inject antibodies? That's something that you
Right. So if you're an old patient, you get COVID, let's say you're in a hospital, you get a single infusion to stop you from getting really sick.
yeah, but then you don't need perfect antibodies, do you?
Who knows? Right. But the
Mm,
point is the current antibodies, the market got pulled from the market because they stopped working, except I
yeah,
think, right. They
yeah,
last for a year. So it's like, well, this is better. I'm like, yeah, but it's already losing it.
yeah. Okay, yeah, it's better, but for a limited time. Yeah, yeah. I guess we need that nice mucosal IgA just so we don't get infected in the first place. That'd be kind of nice.
Yeah.
All right. So, if I may conclude, should I continue? You want to
Yes.
hear about my viral infection story?
Which disease do we have?
RSV. I mean, now there's a vaccine, so will it be relevant? Probably yes, because people don't take vaccines. But so it's a short story, but I think it's nice because it's very translational, and that's why it's published in Science Translational Medicine. Um, First author is Roshin uh, Riley, and the corresponding author is Melody uh, Duval at the Harvard Medical School Children's Hospital, and the publication is called "An Altered Natural Killer Cell Immunotype Characterizes Clinically Severe Pediatric RSV Infection." And when you think about it, it's more of a it's mostly kind of descriptive paper. It's fairly small. Uh, and the mechanism there has a little bit of mechanism, so I think that's really nice. It's not a super large paper, but I just want to say before I start that what I realized, and they also comment this on the discussion, is that doing this kind of work on pediatric patients is really, really hard. You get very little sample. They have these patients that are intubated, they're on ventilators, or they're having mechanical ventilation, or it's really hard to get the samples and get to make the right questions. I have the samples to really answer the questions you want. So I really respect so much what they have achieved in this paper. Basically, what they show is that we know that NK. So they start talking about kind of uh, severe respiratory infections. And we know that RSV is one of the most kind of dangerous infections for you really young children. And the numbers in the US are quite important. There's 2 million outpatient visits, 800,000 hospitalizations, and 300 deaths annually only in the US of RSV infection. And most children, they are fine. So apparently most people get RSV seropositive by the age of 2. So we are all kind of exposed to it very early on. Most children are fine, but then you have this small amount of children that are, severely sick, and some of them even need to get on a ventilator. So, or get ventilated, like non-invasive ventilation. So I've been learning a lot about, type of ventilation. Um, but it can be life threatening. So on the other hand, you have NK cells, and we know that NK cells for this innate lymphocytes are well, not this yeah, they're part of the but they are these innate lymphocytes, and they are uh, very important in general in viral infections. And one of the things that they look into is we know that in other diseases such as COVID, have been like differences in NK populations have been described, and they were wondering what is the involvement of NK cells in RSV infection in children that are in the pediatric ICU. And so they have the first thing they look into is these endotracheal aspirates that they can collect from children that have been intubated. So that's also a little bit of the limitations of the things they can get. So they only can get this very invasive material from very severely sick patients that need to be intubated, because otherwise they don't have access to their lungs, because if you don't have to. And one of the things that are they observe is that within this kind of, this fluid from the lungs, there are more NK cells. And this has also been reported for, I think, COVID. And this is I think it's pretty convincing. And so the question is, so they don't see a difference, for example, on T cells, but they do see more NK cells. So they say, well, okay, what's up with the NK cells? And then they move, and the amount of NK seems to correlate with the severity of the symptoms and the this hypoxemia that these patients are experiencing, and the severity, the clinical severity scores. So there's there seems to be a correlation there. So this makes them very interested in it. So then they have a second cohort of pediatric patients. In this case, they are not intubated. So, or most of them are intubated, so they don't have access to this kind of fluid, again, this ETA fluid, but they look more into peripheral blood. And then, interestingly, in peripheral blood, they see the opposite. They see that the most severely ill children have less NK blood cells in their blood. And they also compare to healthy controls and such. And so basically they see this difference, and they also see a difference in the activation markers of these NK cells. So they have these NK cells are expressing more CD69, and they're expressing less NKG2D, which is one of these receptors that are guiding kind of the recognition of the NK cell for a target. So when they're trying to look into the function of the of NK cells in patients with the RSV, they see that on top of these, they seem to have a reduced cytotoxic function. They do this co-culture with K562 cells, and they see reduced death of the cells. But when they look into the supernatant, there's some indication that there's actually more cytotoxic mediators. This is but again, this data is very small. So they have just 4 donors per condition. Again, it's really hard to get the samples from these patients, these children. But I think I think they're onto something. And interestingly, when they look at they have some in vitro cultures in which they look at the synapses, because they say, well, if they seem to be more, or have more of this CD69 mark, which is kind of more activation, but they have less G2D, which is part of the synapses. What about their recognition? And they're less cytotoxic. How do we put this all together? So one of the explanations would be they're not interacting with their target cells properly, but they're still making cytokines and such. Not cytokines, sorry, cytotoxic mediators, because they don't see any difference in cytokine production. So interesting, they have some nice microscopy images in which they show that there's a disruption of the synapses between the NK cells and their targets. Basically, they show that there's this. They measure, for example, the actin filaments and how they are supposed to be focusing on the immune synapses, and they see that there's lower density in the NK cells from RSV patients. There's a more the microtubule organizing center, which is this kind of marker of the polarization of the cell in the case of an immune synapses, is farther away from the target cell. And this is a measure of kind of how well the cells are recognizing their target. And this polarization that is expected is impaired. So they suggest that in RSV patients, one of the kind of the observations that they do, that they have these NK cells they have more NK cells in their lungs, but they're not functioning as cytotoxically as well. And they might be even having an increase off-target kind of bystander effect, because they are still releasing these cytotoxic mediators, but not to the correct cell, so to say. So I think it's very nice, very important to understand, these mechanisms in actual human patients and not only mouse models. But it's really hard because most of these experiments are with very small samples, very few patients they can get. But I think it's what they see. I think is pretty interesting, and I definitely believe that there's a important NK component in the severity of the RSV infection.
So one of my favorite, I don't want to say favorite, but one of my core memories from residency or med school was treating an RSV baby who was not quite intubated, but was in the NICU. And taking care of them. It's one of those core things that you just see and realize how hard the disease is.
Right. Yeah.
Yeah, they're very sick. So only having, you may be biased towards worst case scenario, but I think it's interesting NK cells keep getting implicated.
Yeah. Always there.
Mhm. All right. Well, we're going to be speaking to Doctors JoAnne Flynn and Stephen Hedrick in just a moment. But before we get to that, if you're enjoying the interviews on this podcast, we invite you to read more interviews on the STEMCELL Technologies website. The Immunology Profile Series, immunologists tell their stories, discuss their research, and voice their thoughts and opinions on current topics in immunology. You can find these interviews at www.stemcell.com/immunoprofiles.
So for our second part of the episode today, for our interview, we have a very special episode with very special guests. Joining us are Dr. JoAnne Flynn, who is professor of microbiology and molecular genetics at the University of Pittsburgh, and who was president of AAI in 2018-2019 cycle. And also, we have a double interview today. Is Dr. Stephen Hedrick. He is distinguished professor emeritus at the UC San Diego. And we are talking to them in a special episode about a new initiative of the AAI called Immunology Explained, which attempts to bring the immunology concepts and the experts to everyone, help improve the literacy about immunology to the general public. And we have invited our guests today because they have very distinguished careers in immunology, have become their work has to some extent written textbooks, and they are interested also in helping the general public understand immunology with their work, with their written work. And we're going to be discussing their career and also the importance of initiatives such as Immunology Explained for today's world today, and how important it is for experts to reach out. Thank you so much for joining us, Doctors Hedrick and Flynn, to the Immunology Podcast.
Thank you. Yeah. Pleasure to be here.
Thank you for being here. All right, Brenda, you want to start this one off?
Well, I think looking into the careers of our guests today, it's so interesting, so many very fundamental aspects of immunology. So maybe I'm going to start with JoAnne. Thank you so much for being here, and maybe you can talk to us a little bit about your background, your scientific background, because I think your contributions to, for example, our understanding of tuberculosis and treatments and animal models that really actually have helped us understand the disease are extraordinary. And maybe you can also finish up a little bit about your interest in advocating for immunology and science, and where do you see your role on that?
Thanks for having me. I started out as a microbiologist. My PhD was straight up microbiology. I took immunology from Marion Koshland. And at that time, T cell receptors were just barely, being discovered and understanding. So the only thing I knew about immunology was BDJ rearrangement. And it was really scary to me. And so I did my PhD in Pseudomonas aeruginosa, pathogenesis. And then I joined a lab at Scripps, Maggie So's lab, where I was working on using salmonella as a vaccine delivery vehicle for malaria. And I realized that I didn't really know enough immunology to even think about working on vaccines. So I joined the lab of Barry Bloom, another former AAI president, where I started my work in tuberculosis and really delved into immunology. And that was all in mouse systems, just trying to understand which cells, which features were important in controlling Mycobacterium tuberculosis. I started my lab here at the University of Pittsburgh School of Medicine in 1994, brought all my mouse strains with me, kept studying mouse TB until I became a bit disillusioned with the model. And it didn't recapitulate all the features of human tuberculosis that I was interested in, including, for example, people who are infected yet are asymptomatic. And so, most things in life are serendipitous. And I had my next door office neighbor was a primate vet working with somebody who was working on SIV at the time. And I'm like, hey, maybe we should try making a non-human primate model of TB, which had been done years and years ago, but before people were doing like really cool immunology. And a lot of the immunology in non-human primates, macaques, was worked out because of people working on HIV or SIV, actually. And so he's like, okay, and I'd never even been in a monkey facility, so I had no clue. But we applied for a grant and got a grant and designed a BSL-3, which is what's necessary for doing tuberculosis work, and set about setting up a model. And we did set up a really cool model that really recapitulated all the features of human TB. But it was really very, I have to say, it was a little scary because it was a major career leap in which I could have completely failed, and that would have been awful. But I felt like it was really important for trying to really get at the heart of TB, the immunology of it, the pathogenesis of it. And so in fact, I can tell you that our first paper that we wrote, here's a model, it's latent TB, it's active TB, it's everything in between. I got a one-line review that said, we don't need a non-human primate model of TB. And this is very distressing, right? It also sounded like an opinion, not a review, but we carried on. And in some ways, it was a really scary time. We had to use the model to answer questions when we were still developing the model, but we developed a lot of cool things about it, and including being able to do PET-CT imaging so that we could track the infection in live animals over time and really hone in on the immunology and try to understand which cells are important, when are they important, what are they doing, to really try and get at what's important in TB and what leads to protection. That leads us into the world of vaccines, right? Because, we have a vaccine against TB. It's called BCG, given to newborns in most countries of the world.
I have two, yeah.
Yeah, I would venture to say that most people who die of TB were vaccinated at birth. And so we're clearly not doing a great job. Certainly it is protective against the worst forms of childhood TB, but we need to do better. And so a lot of my lab has shifted to models of protection. We actually reformulated BCG into using it in an intravenous vaccination. Which I thought would not work, and it worked remarkably well and protected really susceptible monkeys, gave them sterilizing protection. And so now we're sorting through the ways that actually works. And then we also have been looking at concomitant immunity, which means protection against reinfection. And it looks like it's different in some ways than the protection given by BCG-IV. So I think the non-human primate model, scary as it was, 24 years ago when we started it, I think it's really gained a lot of traction, and that we are now, I think, really good at understanding how to do it, and that we can look at individual granulomas, which is the site of MTB infection in the lungs and in the lymph nodes, and analyze those separately to say, what is the constellation of immune factors? It's not going to be one thing or another. It's really, how do these factors work together to provide protection?
So I just want to say, clarification maybe for those listeners that don't remember. So BCG is usually administered subcutaneously, right?
Intradermally.
Intradermally, okay. Yeah. And that's the intravenous vaccinations are not that common, are they?
Very uncommon, and for various reasons. Obviously, they're not that practical, right? Mm-hmm. And there are also some danger of putting BCG is a live attenuated vaccine. In other words, it shouldn't cause disease, but you don't know what happens if you put it in the bloodstream. Monkeys were fine, but there's also some, concerns about that.
But people in the US don't get vaccinated at all with BCG, do they?
Nope.
No, right? And I think also in Europe, it's not that common either, because, you can tell if you are a South American immigrant because of the scars that the vaccine leaves in your arms. So that's pretty clear. You can
relatively recent thing that European countries don't administer BCG, in part because there aren't a lot of companies that make BCG anymore. But so, I mean, tuberculosis is a disease of
Yeah.
poor people. And as the socioeconomic status of a country rises, the TB rates drop. I mean, it's classical, right?
Yeah.
And part of that is malnutrition, and part of that is access to healthcare. But so certain countries in Europe have stopped vaccinating.
Yeah. And in general, the protection against the lung infection is minimal. So it really protects the children from like the severe type of meningitis that you get from
Or any kind of disseminated TB. Yeah.
So beyond that is very little protection anyway. So
And it depends on the population. There's a north-south thing where it seems to work better in the north than it does in the south. And there are certainly studies, for example, in Alaska, where the protection can last 60 years. And then there's big clinical trials in India with zero protection. So it's a very complicated situation.
We're going to talk more on vaccines in a sec, but I wanted to pull Stephen in here too, because I know he's been involved in vaccine advocacy. But I wanted to start back with your research, Stephen, with T-cell receptors, and then how that, I mean, it may be obvious in immunology, but how that led you to be, pushing on the forefront of vaccine advocacy.
Well, first, let me say that I think Dr. Flynn's career trajectory going to a new model and tackling something as important as TB in the world is just inspiring. And she didn't mention, but she well knows that it's probably killed more people than any other disease in history. Maybe smallpox, is up there, but no, she's saying no. So it's a really important issue in the world, and there are many forms of TB now, or cases of TB where they're virtually antibiotic resistant. So all these things really add up to making an important issue to deal with, and one that doesn't get as much traction in the U.S. as it might deserve. But I started a bit before JoAnne, and my introduction was I was an engineering major as an undergraduate, and somebody who lived upstairs from me said, who was pre-med, said, I go to a journal club on Tuesday nights. Would you like to come? So I said, well, sure. I was actually a double major with biology. And so we would stop and get a drink and then go down to journal club. And the first one I ever attended was trying to figure out how T cells recognized antigen. And at that time in immunology, we only had 3 cells. We had T cells, B cells, and macrophages. And the only thing we really knew too much about was T helper cells that would help B cells make antibodies. And so all the models and everything were focused on that. And I hate to tell you what year that was, but in any event, over the next few years, people made a lot of attempts to find antigen-specific factors or receptors or genetics that would enlighten the field on how T cells were able to recognize antigen. And the background of that is that we had almost 70 years of information on antibodies leading up to Tonegawa's discovery of gene rearrangements that were the hallmark of diversity, receptor diversity in B cells. And yet we knew nothing about T cells. And that was just my passion. I wanted to do something like that. And so, for a PhD, I worked on an immune response to an antigen, and we called it immune response genes that map to the major histocompatibility complex. And, that published things on that, but it still wasn't very enlightening with respect to the T cell receptor. So I found that at the NIH, they were making T cell clones. And I thought that the key to looking for the receptor was to have a monoclonal population, similar to the way hybridomas were important for characterizing antibodies. And I was lucky enough, so I got to the NIH, and then about 6 months later, another fellow came to the NIH, Mark Davis, and he was involved in recombinant DNA research at Caltech. And so we sort of combined our interests in cellular immunology and looking for T cell receptors with the newest techniques of molecular biology. With a lot of input from Mark, we were able to make libraries and screen genes and eventually come up with a gene that was rearranging, but it was expressed in T cells and not B cells. And so that sort of led us on to finding these clones that eventually became that we identified as the beta chain of the T cell receptor. And once that was done, and the alpha chain was then cloned a couple of years later, that really allowed everyone to study T cells. Now, to be honest, we also needed to understand how antigen presentation worked, and that was done by several groups that found that peptides bound to major histocompatibility molecules. So at that point, we had the whole complex understood, at least, if not isolated. We could go on to look at T cell development and lots of other aspects of T cell biology. So that's kind of how I got started in immunology. And I guess I'm interested in a lot of aspects of actual cellular immunology. It's not just T cell development. And so that led me to do lots of reading about the evolution of the immune system and how the immune system and pathogens co-evolve. And then the reason why humans have so many diseases that we have to vaccinate for, and why that should be. And so I spent, I don't know, the last 15 or 20 years thinking about the evolution of the immune system, and that led to my interest in vaccines. So and I'll just tell you one little more anecdote. So my daughter lived in West LA, and she had a very good friend who didn't believe in vaccination. And so I thought, well, this is something I know something about, so I should write a paper on this for people like her. And so for about 5 years, I worked on this paper on the imperative to vaccinate, and finally got it published in the Journal of Pediatrics, hoping that pediatricians could share it with their parents who were worried about vaccination. So that's where we are in terms that. I think that answers what your questions in terms of where I came from and how I got interested in vaccines. At this point, I consult for the Justice Department on vaccine injury compensation program. And so I take cases where there has been an alleged event that's occurred subsequent to a vaccination, and write a report on whether or not that seems to be supported by the science of vaccination and immunology.
I want to say two things. First, we had Mark Davis in the show before, and I am so fascinated hearing like the stories behind how we know all these things about T cells, for example, which are my main cell of interest, and how we didn't know those things, some years ago. And I'm pretty sure our listeners are also going to be very uh very motivated, or very kind of it's just something different when you hear how these things came to be, that we didn't always know this. And I'm sorry, I don't want to imply a lot of time has passed, but I just find it great when we have guests like you who were there when it happened.
Brenda, that's a great point, because when I was in college and I would take classes, I was thinking, well, what am I ever going to work on? Everything is known.
Right?
You don't know where the gaps are until you really delve into it.
Right. And I think there's always gaps, right? Everybody had gaps. And of course, for us, it's very obvious when you see, retrospectively what other people did 20, 30, 50 years ago. Of course, we had to know that. But I think that if anything, it should serve, as you say, as inspiration for, how to find the next thing to work on. And the other thing I want to say is, So regarding this idea of the imperative to vaccinate, and maybe we can get a little bit more on vaccines in general. And I mean, we all know that vaccination is important, right? And I'm pretty sure all of our listeners have no quarrel with that assumption. But sometimes I think it is not, we don't realize actually how much and how razor thin the margins sometimes can be. And I think we have the example particularly of of measles, and how having even minute amount of unvaccinated population can completely or can to some extent really eliminate the protection that the communal protection that vaccination gives. I think the numbers are 94%. You need 94% vaccination, like protection, in order for the whole population, whole community to be protected.
Or measles.
For measles, you're right. Yes.
It's different. It's probably different for each infectious disease.
Yeah. No, I meant measles. Yes.
Measles
So
is the most contagious, right?
exactly. So it's probably, the most extreme example, but this is nowadays, I think we've hearing it so often, right? There's this outbreaks that happen in all over, in the US, also here in Germany, for example, there's large populations that are having this issue. So my question to both of you, as advocacy agents, as scientists that have an interest, what are the important functions that a scientist can have, and that you have, or maybe your listeners can consider when thinking about explaining immunology to their non-scientific peers, or their family, or their community? Or what are the kinds of specific actions or roles in their community that they can take in order to help in the community understand, for example, vaccination? There's something that requires an action. You have to convince people to do it. You are helping in committees. You're in particular, you are contributing to governmental agencies, or kind of public discourse. But maybe our listeners have at the moment more modest contributions in mind. What would you say? I don't know if maybe JoAnne, you wanna start.
Let me start with just immunology, right? So, what I find really interesting is when I was on AAI Council, we would always go up to the Hill, Capitol Hill, and talk to our representatives or their staffers. And at first, it was really hard. They're like, immunology? And we're like, but immunology underlies almost every disease, not just infectious disease, but almost every disease. And when immunotherapy for cancer became a thing, it was so much easier for us to talk about immunology, because we could say, so anti-PD-1 therapies were not, people discovered PD-1 because they were studying T cells and understanding how T cells were regulated. It wasn't like, so we can make some great cancer therapy. It was like basic research on T cells. And from that, you get things that are good for, treating cancer. And it became so much easier for us to explain immunology when they could understand that there was a point to it, that all diseases eventually have some immune component, because our body is built that way, right? The immunology is important. It can be good and bad, right? But it's important. And so that became easier. And then when the COVID vaccines came out, it was easy enough to explain, here's why we need some immunology, right? So that we can make new vaccines in a hurry when something happens. And so in terms of dealing with the community, when the COVID vaccines came out, I actually put together a PowerPoint for my youngest, was in high school, in a room, but she was in high school, where I actually gave a presentation of, here's how these new mRNA vaccines actually work, and here's the immune responses that they're engendering that can protect against a virus. And I went through beginning to end of saying, here's how most of our vaccines work, which is by antibodies. Here's how these new vaccines work. And I actually then sent the PowerPoint to my mother, who lives in a community out in California that, is a bunch of houses and the over 55 community, and said, share it with your friends because they have questions, right? And I tried to make it as simple as possible, just with the idea of don't be afraid of this. And this didn't come out of nowhere. There's a whole background of people looking at coronaviruses, which is the virus family of SARS-CoV-2, and a whole bunch of research on using mRNA for various reasons. And I think it helped them to understand it didn't just pop out of nowhere. And so everything comes from somewhere, right? And it's really that basic scientific research that leads to advances that treat things like cancer and infectious diseases and autoimmunity. And I think that those messages of understanding the basic research, which both Steve and I have done, is really important for making these breakthroughs that can make a difference in the world based in immunology.
So I had a kind of question to follow up that maybe Stephen can answer here, which is how do we then stop throwing the baby out with the bathwater? What by that I mean is, hey, measles vaccine, amazing. Hep B, sterilizing immunity. First rounds of COVID prevent you from dying, right? And then on the other hand, you have huge resistance to annual flu shots, maybe some of these extra 5th, 6th, 7th COVID boosters, which do have less data. But then someone's like, well, I'm not going to do that. So I'm not going to do any vaccines. They're all bad now. Like you don't go from, well, maybe this one, like you're kind of on the fringe of the data here. It's kind of a personal choice. Fine. But then you don't just stop there. The response is, don't vaccinate my kids for anything anymore. It's like you getting that granularity seems tough. So Stephen, when you were talking about the imperative to vaccinate and the work you're doing, how have you been able to help people with that resolution that's sometimes needed? Because not every drug is equal. Not every vaccine operates identically, so on and so forth.
Well, the problem that I think we have as a culture right now is that there's an issue with authority. And so everybody has a way of communicating with the public through various social media outlets. And it's very difficult for people to sort through that information. We have a glut of information, and most of the information that you get, I hate to say it, is garbage. Not necessarily just in vaccines, but in everything. I mean, there's just too much information, and it's difficult to sort through. And so I thought the way I could contribute was to put out something that I thought was scholarly and hope that it could get a little bit of traction. Now, it doesn't, compared to any TikTok presentation, it just has no readership. And so to answer your question is, I think it's difficult. I think finding an outlet, finding a way to reach lots of people, especially in something as technical as vaccination, is difficult. I hate to be a naysayer, but I haven't really found a good way to communicate with lots and lots of people in a nuanced way. As you point out, you can't just look at it as good or bad. Every single drug has problems. And the way I talk to people about this is I say, you're born with a danger of all of these diseases. There's no completely safe haven against infectious disease. Your best bet is to get vaccinated, because all of the being infected are much, much worse than the side effects of vaccination. So that's a general statement, but then when you bring up the idea of annual influenza vaccines, or even more difficult, the semi-annual vaccination to COVID, it's not so clear that it's always beneficial to get every single COVID vaccine that's going to come out. I mean, I don't. And so I don't have a great answer for you how to address this, especially when you have politicians that come out, especially several politicians had medical degrees, and they came out saying, oh yeah, you don't have to get all the vaccines, you don't have to get this, or you don't have to get that, or it's a hoax, or boy, when you have to overcome those sorts of issues, it's very difficult. I mean, maybe JoAnne has a better idea how to do this.
I don't, but I tell you, I taught medical school for a very long time, and one of my lectures was vaccines, and I would teach it prior to COVID from a risk-benefit sort of idea. In other words, if people saw kids with diphtheria, for example, right, dying of diphtheria, those parents want their kids not to get diphtheria, and therefore they would vaccinate, right? Same thing with polio. I mean, parents were so afraid of their kids getting polio. Everybody lined up for the polio vaccine.
I was one of the first in line as a child.
Oh, I love that. That's great. And, developed here at the University of Pittsburgh. And so I thought, this made sense, right? If you see that, a baby with tetanus, you're going to think, boy, I don't want my kid to have that, right? And so I always thought that made sense until COVID happened, when people whose family members were dying of COVID still had no interest in being vaccinated. And I agree with Steve 100%. It's very political, and it's hard to fight the disinformation that was out there. And so I think I don't have a good way of doing that. And people during the measles outbreaks that have happened, you can't vaccinate after the fact except for rabies. That's the only one, right? And so when your kid has measles, it's a little too late to vaccinate, right? And measles is particularly difficult because we vaccinate at 1 year, yet babies are still susceptible. Particularly in countries where we don't have a lot of measles, babies are still susceptible before that 1 year time point. And so it becomes very difficult to reach these groups, as Steve said, because they've heard so much stuff that just isn't necessarily true. And the nuances are 100% there. And so you cannot talk about vaccines as black and white. You have to really put it into some context. And I find this extremely, extremely difficult.
I think nuance in general is difficult to get across. But that's, to some extent, it's the whole point, right? It's understanding the nuance will make you do the right decisions, understand what's going on.
I have another thing to point out, and that is that there's lots of reasons to believe that you can't make this entirely voluntary. And there's sort of game theory that I actually discussed a little bit in my article, but there's also just the idea, and the ideas that we're bringing up right now is that people can't do this on their own. They can't make these decisions on their own when it affects the whole community. And so, for instance, California, they passed a law that kids going to school have to be vaccinated. And there are a few exceptions, but it's only for medical reasons. It used to be for, religious or philosophical reasons. And in principle, they don't allow that any longer. And so it's made a big difference in getting communities vaccinated. On the other hand, Oregon used to require vaccination, and they went the other way. And all you have to do, at least last time I looked, all you have to do is go online and fill out a form that says you don't believe in vaccines, and you don't have to get your kid vaccinated. And so now there are kids living in many, many communities that have very low levels of measles vaccination. So I really think that it almost, I hate to say this, I sound like a fascist or something, but I have to say, I think it has to be required.
Yeah. I mean, seatbelts. Exactly. Seatbelts are required.
You have to wear seatbelts. You have to have a driver's license. You have to be 21 to drink. Legal.
Right. I mean, seatbelts protect you, but the driver's license protects the community. and the vaccine is protecting the community. You just don't want to have this rampant disease going around the country. And as Jason said at the beginning, vaccines were necessary in order for people not to die. Now that most people have been exposed or had a vaccine, it's very unlikely that you're going to die from COVID. But at the beginning, it wasn't. And I did read a lot of sort of stories of people who didn't believe in COVID or the vaccines, and then a loved one died, and they were converted almost immediately. So, these things do happen. I mean, it's hard to deny when somebody close to you actually dies of COVID. So I don't know where we stand now. I mean, as I said, at this point, most people have some immunity, and so they're probably not going to die. And whether or not it's important for everyone to get every vaccine is up for debate.
Every COVID vaccine.
Every COVID vaccine, excuse me.
Right. Yeah. I mean, exactly. That's the whole point. People are saying, well, I mean, you don't need to vaccinate against COVID anymore because really it's not necessary. Then why would I vaccinate against other things? Probably all the same. But again, nuance. It's so difficult to transmit the nuance. Not for nothing, immunologists, we got to learn for so many years until we think we are kind of understanding what's going on. And I think there is this frustration in general that you want to understand things easy. Everything has to be kind of brought down to certain levels and some things, certain concepts are just difficult. They're just complex. And that complexity needs to be maintained because that is the essence of how these things work. If it were easy, we wouldn't be having this discussion.
Yeah. I led an initiative to make sure that students at UC San Diego had to take a philosophy course called epistemology. In other words, how do you know what you know? And most people at this point think they know something because they, read it someplace,
right,
and without checking the source and the veracity of the source. And it's an old philosophical issue of how you know what you know. And I thought if everyone had to be exposed to that, at least at the college level, that would help the education of the populace.
That's a very interesting angle.
I love that. So to wrap up a little bit on Immunology Explained, I think this will end up probably being one of our final questions. We talked about how, vaccine nuance is tough. Have you found with some of the other stuff you've done with Immunology Explained or other areas that the nuance is transmitting better? I mean, everyone has their own personal experience with vaccines, which probably makes it a little harder, right? You have your inherent cognitive biases. Have you guys found better access with autoimmunity, aging, and any of the other axes that Immunology Explained in the work you guys have been doing? Anything that's been able to gain a little more traction?
Well, I think autoimmunity is just as hard as vaccine.
That's what I was going to say. So many people I know, including me and everybody in my family, have some form of autoimmunity, and people come to me and say, well, what's this about and why do I have it? And boy, that's a hard question.
Right.
Yeah. I think, again, it's not one thing, right? I think getting across the concept of balance in the immune system is one that is really, again, very nuanced, right? Because that balance means different things in different situations. But just like any part of your body, something can go in one direction too much and the other direction too much, and neither one of those is good. And our immune system is remarkable at balancing, right? But it also faces a lot of external and internal threats that can tip it in these different directions. I don't know. I think autoimmunity is a very tough one to explain because there's so many versions of it, right?
And evolutionarily, we were subject to acute infectious diseases for the last several thousand years as human beings. And so there were certain alleles that were promoted and selected for, had a selective advantage that also now cause autoimmune disease. So it turns out humans are particularly sensitive to autoimmunity, more so than most wild animals. I mean, there's autoimmunity you can find in domesticated animals, But in wild animals, I don't know for a fact, but I imagine it's quite a bit reduced compared to human beings.
That's a good point, Steve.
Mhm.
And I guess also the aging of our immune system, which is another topic from that is covered in Immunology Explained, is also very unique to our experience, and the way that we age and our immune system determines, to some extent, has a great influence on the way we age and our experience with pathogens, and it also kind of conditions our immune system in our old age. And
Well,
I
not
think
only
there's
that, but most of the diseases of old age have some immune basis, some inflammatory basis.
exactly, yeah.
And we keep ourselves alive because we're human beings um, a long, long time with medical interventions when, our physiological age has gone to its max, so to speak.
And the vaccines mostly don't work as well in older people because the immune system is kind of going downhill, right? So that's really important as well. I mean, some of the vaccines for older humans have been modified to be, give a stronger immune response in a person whose immune response has waned.
Yeah, the seasonal influenza is the good example of that. I think it's about 4 times as high a concentration of vaccine as for
Oh.
younger people.
Oh, so there's a special dose for older people. That's very interesting.
And then there are some people who acquire a huge percentage of their T cells that are specific for cytomegalovirus,
Yes, CMV,
and they
yes.
tend to have a poorer outcome for infectious disease and all-cause mortality as well.
Well, on that very bright note, because CMV is like at least 50% of any population has, and if you're lucky, it's 50%, and other countries have like 80%, 90% seroprevalence. So
I dodged that.
yeah, you're negative, you're seronegative, good for you.
They had to check because of my colitis. When I went on anti-TNFs and 6-MP and all that, they had to check, and I am CMV negative.
Oh, good for you.
If you're on anti-TNF,
And
you're more
EV
susceptible to tuberculosis.
Yeah, not anymore. I went to Entyvio, thank you. But but that was a whole thing for a while. But I was on a TB ward on anti-TNFs as a med student.
Oh,
Wow.
great.
Yeah, that was an OSHA issue. Thank you, N95.
But so I think that's, well, we could talk so much about the aging immune system, all those infections that we accumulate through life, but I think that would be a completely separate episode. We are reaching the end of our conversation, and I just want to thank you, Doctors JoAnne Flynn from the University of Pittsburgh, and Stephen Hedrick from the UC San Diego, for joining us today, for talking about their research and also the initiatives of the AI, such as Immunology Explained, trying to bring immunology education, knowledge to the wider public, and also in your work, policy generation, like government, helping the people that are making the decisions understand what's going on. I find that such awesome work, and I think we should all be doing it in one level or another, even if it's just to our community, or if we are lucky enough or we have the resources to reach out a larger community. That's super important. So I'm very
I love that. I love the new public facing AI. I really think
yes.
it's so important. And I love your idea though, Steve, of ask an immunologist, right? It's
Yeah,
great.
it'd be fun, I think. I have to point out that I ran for Immunology Council twice and lost. So my hat's off to you, JoAnne, for being able to get through.
It was an experience.
Yeah, sure it was.
I love
Yes.
AI.
Yeah, so if that's an interesting idea, well, I'm sure there's lots of people who would be interested, immunologists interested to participate in some sort of a forum. I mean, an ongoing online forum like that.
Yeah.
Probably looking at a couple of them right here.
Yes.
Well, thanks for having me. I really loved it.
Yeah. Thanks for coming. Thank you so much for joining. And let's see what else comes into the AI Immunology Explained website. And our listeners, if you have family, friends, I also think they even have a version in Spanish. So that's a great place to find material for reaching out to your community.
And if anybody's interested in this article I wrote on the imperative to vaccinate. Maybe you could link it.
Yes, we will include a link in our webpage for this podcast. We'll make sure that we're going to include it in our social media so our listeners can find it easily. It's a great article. It's a beautiful article. So I absolutely recommend everyone to read it.
Thank you.
Thanks for having us. It's been my pleasure.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.